We have already discussed basic autosomal patterns of inheritance in Chapter 7. Chapter 8 goes over some of the more complex patterns of inheritance as well as DNA fingerprinting. For some genes, two identical copies of a dominant allele are required for expression of the full effects of a phenotype. In this case, the phenotype of the heterozygote is intermediate between both homozygotes. This situation is called incomplete dominance. In humans, curly hair is incompletely dominant to straight hair. In a situation where one parent has curly hair and one parent has straight hair, the offspring would likely have wavy hair. Another example of incomplete dominance in humans is familial hypercholesterolemia. If you have two abnormal alleles, you have severe symptoms, including heart attacks as a child. If you have one normal and one abnormal allele, you have moderate symptoms that can lead to heart attacks as a young adult. If you have two normal alleles, you don't have any symptoms associated with this disorder. In codominance, there is a situation where either neither allele is dominant to the other, or several alleles are dominant at the same time. If neither allele is dominant, then both are fully expressed in that individual. If several alleles are dominant, they both express and will mask the expression of the recessive allele. In multiple allelism, more than two alleles control a trait. However, even in multiple allelism, each individual still only has two alleles for that trait. The human ABO blood types are an example of both codominance and multiple allelism. In the human ABO blood system, there are three possible alleles that can determine your blood type. Human red blood cells can produce certain sugars on their surface. These sugars act as an antigen. An antigen is a type of molecule that stimulates the immune system to create antibodies. If you have the dominant A allele, which is indicated by an uppercase I with an A superscript, then your blood cells make sugar A, which could stimulate an immune response if given to someone who does not have any A sugars on their red blood cells. The person who receives this blood would mount an immune response, an immense immune response, and this reaction would create a lot of antibodies to the A sugar, and then your immune system would try to destroy the foreign red blood cells as quickly as possible. This could lead to clots in your vessels or even an anaphylactic reaction that could be deadly. The human ABO system is also an example of codominance because both the dominant alleles, the I, uppercase I with an A and the uppercase I with a B, are fully expressed or are fully dominant in the presence of the other. There are four possible phenotypes for this system. You can have type A, type B, type AB, and type O blood. To have type A blood, you could be either homozygous dominant for the A allele or heterozygous with the A allele. To have type B blood, you could be either homozygous dominant for the B allele or heterozygous with the B allele. Type AB blood is the example of codominance because if you have both of these dominant alleles, you make both the sugars or both the antigens on your red blood cells. This is why people with type AB blood are considered to be the universal acceptors. Because they make both types of antigens normally, they aren't going to have an immune response to any type of blood, so they can safely accept any type of blood. People with type O blood have both recessive alleles, so they don't make any antigens at all. Their red blood cells have no antigens. They are considered to be the universal donors. Because they don't make any antigens, their blood is safe for anyone to accept. So in an emergency situation where there isn't time to do a blood typing test, the health staff will typically use type O blood whenever they can. Okay, so let's look at a Punnett square crossing a person with type O blood and a person with type AB blood. The parent with type O blood is shown on top of the square by the two recessive alleles that are indicated by the lowercase i's. The parent with type AB blood has both dominant alleles, so an uppercase I superscript A and uppercase I superscript. Now we've combined those alleles to see what genotypes the children could possibly have. Out of the four possible combinations for offspring, all four are heterozygous, meaning they have two different types of alleles. Two are heterozygous with the dominant A allele, and two are heterozygous with the dominant B allele. the possible phenotypes of the offspring would be half type A, so 50% type A blood, and half, or 50%, type B. You do need to understand how this blood system works, and you should be able to do Punnett squares involving the blood types. Another type of inheritance involves pleiotropy. Pleiotropy occurs when a single gene affects two or more distinct and seemingly unrelated traits. Typically, when we talk about pleiotropy, we are talking about mutant genes or abnormal genes. Your textbook gives the example of hemophilia, which is a clotting disorder that's relatively common in certain royal families but is fairly rare in the rest of the population. Another example is Marfan syndrome. People with Marfan syndrome have long arms, legs, hands, and feet. They also typically have poor eyesight and a weakened aorta. These characteristics are all due to the production of abnormal connective tissue. They're also characteristics of good basketball players, well, minus the eyesight and the aorta issue. But because of this, some schools have begun screening basketball players for this disease. Undiagnosed syndrome could lead to sudden death due to a ruptured aorta. So clearly these people should not be straining their hearts through intense exercise, which means they really shouldn't be playing basketball, despite the fact that their size and shape lends them towards being excellent at it. We've primarily talked about autosomal inheritance patterns so far. Remember, your autosomes are all the chromosomes that are not sex chromosomes. Now we're going to talk about inheritance patterns that involve the sex chromosomes. Because the Y chromosome is so very tiny and really doesn't do much other than determine testosterone hormonal production, it's not really very interesting as far as inheritance goes. The X chromosome, on the other hand, does carry a lot of traits other than just gender and hormonal determination. The term X-linked is used for genes that have nothing to do with gender but are still carried on the X chromosome. Because females have two X chromosomes, they are less likely to be affected by genetic diseases that are carried on the X chromosomes. Even if one of their X's is abnormal, the other one can be normal, and that would prevent them from displaying a recessive disorder. For a female to have a recessive X-linked disorder, they would have to have two abnormal X chromosomes. However, for a male to have a recessive X-linked disorder, he only has to have one abnormal X chromosome because that's all he can have, at least. He'd have one X and one Y. Women with one abnormal and one normal X chromosome are considered to be silent carriers of recessive X-linked diseases because even though they don't display the disease, they could pass it on to their male children. Color blindness, muscular dystrophy, and hemophilia are all examples of X-linked recessive disorders. About 8% of Caucasian men have red-green color blindness. Most of these see brighter greens as tans, olive greens as brown, and reds as reddish browns. This Punnett square shows a cross between a normal father and a mother that is phenotypically normal, so she appears normal, but she carries a recessive X-linked disorder on one of her X chromosomes. This could be color blindness, kind of in this example, or it could be hemophilia. When you complete the Punnett square, you can see that out of the 4 possible combinations, 2 are male and 2 are female. So half have 2 X chromosomes, and the other half have an X and a Y chromosome. Of the females, half of them will be completely normal, both genotypically and phenotypically. The other half will be phenotypically normal, but will be carriers for the disease because of one abnormal X chromosome. They don't show the disease because they've got a normal one, but because they have an abnormal X chromosome, they're carrying that disease, and they could pass it on. Of the males in this Punnett square, half will be genotypically and phenotypically normal. The other half will have the disease, whether that's something as minor as color blindness or something as major as hemophilia that could actually shorten their lifespan. So 1 out of the 4 children will actually manifest the disease. 1 out of the 4 is a carrier for the disease, and then the other 2 out of the 4 are normal. A pedigree is a family tree that follows the inheritance of a genetic trait for several generations of relatives to try to determine how it's inherited. In this example, the squares represent males and the circles represent females. The black spots are the family members that have the trait. In both pedigree A and pedigree B, the trait shows up fairly rarely, so it's most likely a recessive trait. The last thing I want to talk about from this chapter is DNA fingerprinting. This is a multi-step process. In the first part, you take a very small piece of DNA that you've got on hand, and you amplify it, or multiply the amount you have, through a process called PCR. PCR is actually used in many different types of tests, not only to amplify DNA, but also to amplify something like viruses, if you're checking a blood sample for a specific type of virus and want to know if it's present, even in small amounts. PCR, for the purpose of DNA fingerprinting, targets specific sections of DNA called variable number tandem repeats, or VNTRs, that are unique for different individuals, just like the whorls on your fingertip are unique for different people. These amplified fragments of DNA generated by PCR are then separated from each other through gel electrophoresis. In gel electrophoresis, the fragments are put into distinct clumps at one end of the agarose gel, which has a consistency very similar to gelatin. An electrical current is applied to get the fragments to move across the gel, and they end up separating out based on their weight and their charge. How they separate out will be different for each person and can be read as a banding pattern, which you'll kind of see on the next page, and that's pretty unique to each individual. Here's a diagram of what gel electrophoresis looks like. You can see the slots containing the DNA samples at one end of the gel. As the electrical current is applied, the DNA will travel down the gel and then deposit out in different locations depending on how big it is. So the larger pieces will settle out first because they're heavier and they don't travel as far as the shorter pieces. The banding pattern that results from how these DNA pieces settle out is almost completely unique for each person. Here are some genetic practice problems that you can work through on your own at home. By the time you get done with this chapter, you should be able to do all of these problems and discuss the answers and kind of understand how those answers occur. So make sure you work through these, and if you have any questions about them or if you can't get through them, feel free to email me about that.